All right, if there aren't questions, then what we're going to do is move on and we're going to talk about mRNA processing. So we've talked about transcription.
Is this still chapter 1?
I don't remember what chapter we're at. So it's the one where we're talking about transcription. But, so transcription is the process, so just to remind us, what we're talking about when we do this, we're doing nucleotides, most of it's nucleotide, but we're going to talk about translation as well. So we're talking about, we've got DNA, goes to RNA, goes to protein, and so far we have talked about DNA replications of DNA making DNA. We have talked a little bit about using RNA to make DNA, so using an RNA template. We talked about telomerase making telomeres. We have not, that's a reverse transcription, okay? And then right now we're in this process of talking about DNA going to RNA. And there are a lot of different types of RNA, and we'll talk about more of them a little bit later today. We're not going to spend too much time on various types of RNA. But in terms of, right now we're talking about the RNA that gets used as a template to make a protein. So transcription is going to make an mRNA usually. Right, you can transcribe DNA to make any of these particular types of RNA, but so transcription can be used to make mRNA, which is the, or let's say that is, I think is mRNA. That is the. I've had this whole which versus that thing explained to me many times. I've never gotten it. So, but the so that is the template for making proteins. All right, so this is the photocopy, I like to say, of the DNA, which is your really perfect copy. So then mRNA has three major processing steps that occur to it before it can actually be used to make proteins. Okay, it's used as a template by the ribosome. And so the first part of that is a 5' cap is added. Okay, the purpose of this 5' cap is to prevent exonuclease degradation of the RNA. All right, so we don't want that to get chewed up. The mRNA is not going to permanently stay around in the cell like the DNA does, but you want it to stay around for a certain amount. So what's interesting here is, well, we get this cap it's one of these 7-methylguanosines. So modified guanosine. Okay, and what's interesting about it is it's attached by this 5' to 5' triphosphate linkage, right? So this is weird. We don't really see that, right? Usually we see this sort of thing. A 5' to 5' triphosphate linkage. So it's a 5' to 5'. So apparently having 3 phosphates in a row wouldn't be enough to stop the exonuclease. I don't know. I don't know what the evolutionary reason for this is. So that's one of our things that happens, right? We're processing the mRNA, so that protects it. The next thing that happens that we're going to talk about is, this is about, there's multiple things going in this figure, but at the 3' end, you're going to add a poly A tail. Okay, so actually before that, the mRNA is cleaved somewhere. And a poly A tail is added. And that poly A tail can be, I'm trying to remember if it's 120 nucleotides. I'm going to just say 100 to 200 nucleotides. In mammals. And here we are talking about higher eukaryotes, okay, rather than bacteria. They have similar sorts of processing, but we're going to talk about splicing of RNA, and that doesn't really happen in bacteria because bacteria are really efficient with their genome. But so this example here shows this stuff. If I have you ignore the intron part, okay, what's going to happen for the moment? I want you to ignore it. We're not going to ignore it for very long. Oops. Okay, so we have this albumin gene here, and it's going to get cut here for this gene. So pretty much close to the end. We're going to lose that piece, and then we're going to add a poly A tail. Yes?
Can you scroll just a little bit?
Mm-hmm. So what I want you to focus on from this figure that has a lot going on is we're going from here to here where we have the poly A tail. But that cut could happen elsewhere, and we'll see that. So you can get different mRNA transcripts from the same gene based on where that poly A tail cut happens. I'm going to say mature mRNAs.
Where's the specification of mature versus?
Mature is going to be after we've had these 3 processing steps happen. Okay. So mature mRNAs, so let's say after the 3 processing steps.
What's the third one?
Intron splicing.
Oh.
So it's based on where the mRNA is cleaved for the poly A tail. All right, so this kind of breaks down this. There was for a very long time there was a one gene, one product hypothesis. In other words, each of your, it's about 20,000 genes, I think, is where they are now. It's hard for me to remember because I went through when they were trying to figure out how many there were. I worked for a company that really hoped that there were lots more, and then it came down to 30, and maybe now it's 20,000. But 20,000 different genes that make more proteins than that, because some of them can make more than one protein, let's say. Okay, then our final step here is we're gonna have what's called intron splicing. All right, and so in eukaryotes, there are intervening sequences in genes that don't code for anything. Let's say that don't code for proteins is probably the best way to say it.
Do these happen sequentially, or are they all kind of happening at the same time, just describing them sequentially?
I don't know.
Cool.
So I don't know if they happen sequentially or not. Yeah. So there are intervening sequences that don't code for proteins. And I didn't want to say, I said they didn't code for anything. But, you know, if you believe that natural selection exists for a reason, there's a couple ways to think about this. You can think about, you know, well, we're big organisms and our DNA metabolism doesn't take much energy compared to everything else. And so we can afford to have a bunch of other DNA in there. Okay, there's some thoughts there. There's some thoughts that our genome is just loaded with viral deactivated viruses. So if somebody gets a virus infection, over time your body learns how to deal with it. The sequence of DNA that got inserted into your ancestor's genome gets deactivated in some way. So it no longer codes for a virus or effective virus, but the pieces are still there. There's a lot of thoughts here. They have found lots of long-range interactions in terms of transcription. So we talked about having a promoter, but you can have something way, 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 way over here if we wrap the DNA all around that can actually come in and influence transcription. And then my research with these folks down at VT, involves ribosomes and stuff, and there are these active and inactive parts of the genome in terms of chromosomes that come together where they're not next to each other on the chromosome. So you can have the chromosome be, if we think about the chromosome doing, let's think about this just being a super long piece of DNA, right? That you end up with, I drew it like this, but this stuff's active, and this stuff's not active. As is the marker, that stuff is not active. Okay, but there's a lot of really fantastically complex stuff, and a lot of it is the subject of ongoing research. All right, so, but eukaryotes are these intervening sequences that don't code for proteins. These sequences are spliced out, and the coding regions are spliced together. Oh, I don't need a purple.
I have like
Is that yours?
12.
Okay.
So
you don't need to support, the chemistry department can buy that. Okay.
I just keep them with me.
These don't code for anything. Or these do code, but this is super active, right? You can think about some genes that you need to make more, you need to have more active, like actin, right? Actin's the cytoskeleton, there's lots of it. When I was in industry, I was doing 2D protein gels, and you'd always have this giant actin splot. You didn't care about the actin splot because it doesn't have anything to do with cancer. You cared about the tiny little splots that might be around it. And then you have regions that you don't have very active, right? Your cell cycle signaling proteins should not be very active. They should just come on for a little bit and go off. But so when eukaryotes are these intervening sequences that don't code for proteins, these are spliced out, and the coding sequences are connected. Okay, and so we have names for these. Exons are the coding sequences. And then introns are the non-coding sequences.
For the purposes of this class, are we just going to say non-coding period or like non-protein coding?
I think non-protein coding is probably better. So the exons are the protein coding. I was just thinking about that myself, that it might be confusing with coding and non-coding strands, which have I talked about in this class, or did I write it in notes and not get to it? I have talked about it?
It sounds vaguely familiar.
It would have been last lecture.
I think so.
Let's make sure I talked about it, because if I give you like a sequence and expect you to tell me which is the coding and which was the
We did
non-coding.
mention specifically that we needed to know that, but I think you called it like the template and non-template strings.
There's template, non-template, and coding and non-coding. They go together.
Yeah.
I don't think I use non-template. Okay, great.
But I remember you specifically saying that if we saw like that on a test, we should be able to
Great.
differentiate.
Okay, so the introns are the protein coding or the non-coding. We talk about as protein sequences. Okay, and so just like above, depending on how introns are spliced, you can keep some of the exons or not all the exons. Okay, depending on which exons are kept. You can get a different protein sequence out of it.
How do the exons connect?
How do they connect?
Yeah.
You're going to splice them together. It'll have something with ligase ability.
Okay.
So you cut the piece apart, they're being held. I don't actually, it's called a spliceosome. There's a set of protein complexes called the spliceosome that will bring them together. And I don't really remember how it works. I mean, I think in one of the books it shows these reactions happening. But it's really, you're going to bring the ends together and then have them do that. And it's an RNA, and so it's a single strand, right? But you can have the 3' end of you'd have the 3' end come in and kind of invade. Sorry, I haven't had people responsible for mechanisms, so I don't even have it in the lecture notes anymore. Different protein sequences can result. After translation. Okay, so you're going to have a different mRNA result that's going to turn into a different protein. And so here's an example of that. I don't know how many of you get migraines. I've had a lot of students, a lot of my Gen Chem class gets migraines. But this is kind of related to this. So we've got this primary transcript that has 1, 2, 3, 4, 5, 6 potential exons that could be spliced together. All right, and so in the thyroid, what's going to happen is we'll get cleavage of this. So what happens? Our poly A site ends up there after 4. Okay, as opposed to in the brain, the poly A site is after 6. So we're going to lose a lot of this. In the thyroid, we'll get the splicing. We'll end up with exons 1, 2, and 3. So here we have the mature mRNA. So if we're going to look at this transcript right here, Bridget, in relation to your previous question, that cap's already on, right at the beginning of this. And then our cleavage, it looks like this would tell you that we poly A cleave and then we splice, if that's the order. We believe this, how much we believe this figure. And then you end up with calcitonin, and this is a hormone that regulates calcium concentrations. Okay, and this other thing over here we have in the brain, we have various splicing. So now we end up with exons 1, 2, 3, 5, and 6, and this is something called CGRP. I didn't look up what it meant exactly, but this is a factor that's involved in migraines. Migraines have to do with blood pressure, so this must be signaling the dilation of blood vessels somehow in the brain. So migraine is when blood vessels dilate, you get extra pressure on your brain for that. And this is actually a migraine drug target. Okay, try to get rid of this thing or not have it made. But both from the same gene. So all your cells have this gene, this is the transcript, but have the gene that codes for this transcript, and then you can make different products depending on what cells you're in. Questions? So those are our three processing events that happen to mRNA. So we have the 5' cap, we have cleavage, and then polyadenylation, which can give us multiple transcripts for the same gene. And then we have our intron splicing, which also can give us multiple transcripts. With that, if there aren't questions, I'm going to move on. I want to talk a little bit about retroviruses.
You said there was two?
What?
You said three, but it was like you said the 5' the cleavage, the poly, and then also the intron splicing.
Yes, and the last two. So the cleavage and polyadenylation is 2,
Okay.
and then 3. The third would be the intron splicing.
Okay, thank you.
The cleavage and the cap, I meant the poly-A tail are one step.
Yes, where you cleave for the poly A tail.
Okay.
Okay, so let's talk about retroviruses. All right, so retroviruses, the big focus over my lifetime with this has been HIV, okay, which causes AIDS. So we've got it's a retrovirus, and it has 3 particular proteins that are really important drug targets. Okay, retroviruses. So an example is HIV. We've got 3 important proteins or enzymes. They are protein enzymes. And this is really in terms of thinking about drug targets, but they're very important for the life cycle. So we've got a protease. We have an integrase. And we have a reverse transcriptase. I'm going to go ahead with reverse transcriptase. And they're really important because the cell doesn't naturally make these sorts of things to help the virus out. So what we've got here is, if we think about the life cycle for this virus, well, the first part of the life cycle, is we've got a virus that's coming in. It has an RNA genome. It's going to take that RNA genome and it's going to bring it into the cell, and we're going to have reverse transcription. This is one enzyme. Okay, this is done by that reverse transcriptase. And you'll see we have multiple steps here. We're going to talk about that in detail, but that enzyme has 3 different functions. Okay, so it's going to go and it's going to take that RNA, it's a single-stranded RNA, to a double-stranded DNA. Let's just point that out, single-stranded. Okay, and then down here, this is double-stranded. Okay, and then that double-stranded DNA is going to get integrated into the human genome. Okay, it gets integrated into the genome of the cell. And then our protease, we'll see what that's doing in a minute. All right, but do you see that before, if a eukaryotic cell does not have a reverse transcriptase and it doesn't have an integrase, the virus can't even get started because it can't access that machinery. It goes into the cell, it shoot in its genome, and then nothing else would happen. You guys see that? Okay, and then the protease is the same thing. So if we come down here and we look at what goes on with this gene, So we have right here, we have a genome for the virus. It has these various genes that produce really big proteins, okay? And those proteins have all the pieces in them, but they're one big piece that's not active. All right, so we have, once we've had reverse transcription, right? We've had integration, all that's happened already. So integrated viral gene. I'm going to say genome, actually. It's more than one gene. And we get, these are inactive. And they need to be cut up into their components to become active, right? So we need to have a protease come in and cut them. And this is that viral protease.
So protease happens after the integrase?
Yes. Okay, and so when you look at this sort of thing, so if the virus manages to get its genome in, right, it's carrying a reverse transcriptase, it's carrying an integrase with it, it integrates its genome in, and then this transcription is just normal cell activity, okay, using your normal eukaryotic, normal, in this case, if we're talking HIV, human RNA polymerase complex. We're going to get the primary transcript that's going to get translated by the ribosomes in the cell that are there, the cell's ribosomes. But then we make these giant polyproteins that can't do anything on their own, and they need to be cut up by a specific viral protease. So the virus has to have the viral protease in it as well to get the ball rolling. Okay, and so that it can make more. And then at that point, it's making its own integrase protease and reverse transcriptase and can keep going. And then these proteins are structural, really, the envelope and the structural proteins. Questions? And so what this means is, if we come back up here, these are drug targets because they are unique to the virus and they're in the virus. Let's say good drug targets. And if you've read, or I don't know, depending on how much you know about HIV or what goes on with AIDS, is that the virus is targeting its T cells, so it's white blood cells that kind of direct things in the immune system. And there's kind of this fight where the virus is, the immune system's recognizing and getting rid of the virus, but it's a back and forth, and it's attacking the immune system. And over time, with no treatment, in almost all people, the virus wins. Okay, sometimes takes a long time for the virus to win in this kind of back and forth fight. And so what happens with the antiviral drugs is that they are taking out one or more of these. Okay, they're addressing more on one of these, and they're slowing the virus down enough that it tips the balance. The immune system can keep it beat down. Okay. Unlike a lot of sorts of things like this, the immune system can't ever really take it out. So you're on the antiviral list for life if you get this. Okay. Yes.
How do like preventative drugs work differently?
How do the preventative drugs? They're probably similar. I'm guessing. I'm not familiar with the preventative drugs, but they're probably similar for that. And there's not. Gosh, I think maybe I've seen things about a vaccine, but I've never seen anything that people are really excited about it. It's the difference between, so when the COVID thing when COVID came out, I was like, yeah, they're not coming up with a vaccine quick for this because they've been trying to make vaccines for AIDS for 40 years or more here, and they haven't successfully done it. But if you think about it, almost nobody who gets the HIV virus without treatment survives. The immune system never wins, pretty much. Whereas COVID, you had almost everybody surviving, right? I mean, untreated COVID, was it probably 99%, 98% survival? And so you've got a case there where you can actually give a vaccine and train the immune system to win, right? That's what a vaccine's about. Whereas you can't train the immune system, you can't help the immune system win if the immune system wasn't going to win anyway. I don't know if that makes sense or not. That's kind of the difference between those two. Now what happens is, we haven't talked about reverse transcriptase. I've got to make sure I get to that. Okay, we're fine. But what happens here is that the virus actually makes, the HIV virus actually makes a lot of errors in terms of this reverse transcription of its genome. It's not very good. Actually, we need to go back and talk about the DNA replication fidelity still. But it doesn't do a good job of not making errors, but it's making lots and lots of offspring, right? And so that's why you've got these viruses that will evolve very quickly in terms of, if you just were to give somebody, you talk about these combination therapies, you're just going to give somebody something that attacked the protease, you'll probably end up with some of the viruses that are made that are resistant to the protease. And so the combination therapies is you're hitting all of these, or at least two of them, okay, simultaneously. And it's hard to get a virus offspring that's genetically changed that it's good against both, or three. Does that make sense? It was interesting. Actually, somebody was talking about this. Whose lab was that? I saw a lab where they were working on HIV. I can't remember if it was a transcriptase or the protease. They were working on one of these. And they were working on trying to figure out how you make drugs that were going to be, the virus was not going to be able to easily make something resistant. So try to make a drug that was going to avoid resistance. But they talked about how you have so much data because someone will get infected, and then what you can do is you can sequence a bunch of the viruses that end up being made, and you can see how fast mutations happen. So they can look at one individual, you can look at one infected individual and just get a whole family of all the different sorts of mutations that tend to happen. And then you can sequence those, and you can use that to then turn around and try and design drugs that avoid those mutations, if it makes sense. If that makes sense. Because not all mutations are going to result in a functional virus. From the scientific perspective, cool stuff. From the health perspective, not so much. Okay, so all right, so let's talk about what reverse transcriptase is. We talked about that in terms of a broader view of retroviruses, but what we're talking about right now is transcription. And so let's talk about the three functions of reverse transcriptase. Okay, so it's a virus. They're similar, sort of. Not one of them, not so much. So the first thing is it can make an RNA copy, or it can make a DNA copy from an RNA template. So you start off with, let's say this is my RNA genome. And this is really the reverse transcriptase, officially reverse transcription. All right, so the first part is going to be reverse transcription. And then, but it needs to get to a double-stranded DNA copy, right? For integration. And so we have this RNase A activity. I'm sorry, RNase H activity. And what that is it's going to, I'll type these out afterwards. But it's going to degrade the RNA template now and just give you a single strand of DNA. All right, and then once it has that single strand, what we're going to get is then you get DNA dependent DNA polymerase activity. So it's essentially replication activity, right? It's going to go ahead and then it's going to make a double strand of this. So all right, does that make sense?
Mm-hmm.
So it does all those things. So it's a pretty multifunctional enzyme. Let's go ahead and write those out real quick. So step one is it makes a DNA copy from an RNA template. Okay, the second part is once we have that DNA RNA hybrid, it degrades the RNA strand of a DNA RNA hybrid. So that's going to be RNA H activity. And then our final activity is to make a DNA copy from a DNA template. And that's DNA dependent DNA polymerase activity makes a DNA strand from a DNA template. Questions on the various activities?
It's like RNA H, and why was the H for?
I don't know why it's called, I don't know what the H is from historically, but it's always, that's the, here RNase H, it's always talking about DNA or degrading an RNA strand of a DNA RNA template. So when we think about if we were to watch, if we'd had the old Flash video, where it for the replication complex and all the different enzymes involved there, one of them was an RNase H. And so it may have to do with what was happening there. It might have to do with, I don't know if they were being cute in that video, but it was kind of H-shaped. But
H for hybrid.
could be H for hybrid, yeah. That's actually a good idea. I like that too. Yes.
When you say these DNA copy from RNA template, any type of RNA, or is it specific?
Well, in this case, it's the viral genome.
Okay.
So it is not, as I understand. Well, I was going to say that it's not just doing it to any mRNA in the cell, but it actually might be. Because people use this. This is really useful in terms of, but I don't know if it's a modified reverse transcriptase. So people use reverse transcriptases to take RNA in a cell. You can take RNAs from a cell, you can make the DNA versions of them, and then you could do PCR. So at the point, because you can't do PCR with RNA, but if you wanted to know what are all things this cell's expressing, you could take all its mRNAs, you could turn them into DNA, and then you could do PCR on that, and then you'd have enough to sequence. Does that make sense? It's something that gets used by various people if you're trying to look at what would be called the transcriptome of a cell. That's how they figure out what cells are making, is that they, at least originally, you would make a bunch of DNA copies and then you would do sequencing on them and figure out what they were. Other questions? Okay. So we're talking about viruses. We're talking about viruses integrating parts of their genome into the human genome. Got Harold Varmus here. He earned the Nobel Prize in 1989 for showing that viruses can cause cancer. Okay, we're going to talk about a couple viruses that do that. I partly put his picture up here because this is one of those Nobel Prize winners I've had conversations with, and I was pretty clueless my whole career. But then found out later he was a Nobel Prize winner. So I should have known better because did I ever tell you the story? Stop me if I've told this story. My buddy Naranjan, who was all over this guy, we were at some meeting talking. He was there, and Naranjan just had to talk to him the whole time. So I should have known because Naranjan was on top of things that he was famous. Anyway, but. So what he showed, and I've got your same picture over here, but this was originally shown in chicken. There's chicken sarcoma virus. Okay, and this causes cancer in chickens. And the way that occurs is it integrates its genome in front of a gene. The gene is actually it's got like an SRC gene here. And what that SRC gene does is that gene overexpresses a tyrosine kinase, which causes things to blow through the cell cycle. So viruses can pick up parts of people's genes, your genes as well, but it overexpresses a tyrosine kinase. Sorry, not kinases, kinase. That causes it signals cells to continue through the cell cycle when they normally wouldn't. Okay, so they're going to phosphorylate things. We'll just say cells to divide.
Is this about the reverse transcriptase, or is this just
This is not about the reverse transcriptase. This is just how viruses cause cancer.
okay?
Okay. Something you may have, so you're not probably so concerned about chickens getting cancer, but you might be concerned with human papillomavirus, okay, which causes cervical cancer. And so what goes on here is there's some oncoproteins in this virus. Okay, they're going to cause cells to divide. And what goes on is I think this blocks P53. So P53 is an important protein involved in various cancers that stops the cells from dividing.
So they make oncoproteins make that?
No, you naturally make that. Let's leave that. Stop cells from dividing. So this is not just involved with cervical cancer, but we've got, and so the virus makes a protein that interferes with this. Okay. And so once again, the cells don't stop dividing. If cells have damage, one of the things that's involved is P53 is supposed to say, hey, don't keep going. And you're no longer stopping the cell cycle because of this. Okay. And so this is how you could have a vaccine. You can have a vaccine for human papillomavirus that's preventing cancer. Okay. Questions? Okay.
Wait,
Yeah, yeah.
okay.
Okay, so with that, we're going to move on and start moving towards translation. We're actually going to talk about tRNAs first, but these are things that get made. Okay. So in translation, so we talk about DNA, RNA, protein then. Okay. Right. So we've talked about this. We've talked about this. This happens. We really haven't talked about it. Actually, if you think about COVID, COVID is RNA to RNA. Okay, in terms of the genome being replicated. And now we're here. Okay, we've talked about that too. But so what we've got here is a tRNA. So RNAs are not just mRNAs. You've got a gazillion different types of RNA. Let's go down to the bottom here. I think that's what this picture is. I couldn't see it this morning. I was trying to remember it from previous years. Still can't see it. I don't know if we're going to get it big enough that we can see anyway. Actually, I can zoom it in and then I'll zoom back out. So this was, I think I went to Wikipedia or something and just went to, hey, give me types of the page on types of RNA. Maybe I went to a couple pages. But these are all the various different types of RNA that there are. And for your textbook, I like your textbook to be cheap. So you've got a textbook that's, 14 years old or something. Biochemistry doesn't move that fast, but where it is missing, lacking, is in talking about a bunch of different types of RNA that I'm not going to spend much time on here anyway. But there are all sorts of places that the cells use RNA here. So we've talked about messenger RNA, right, at this point. Ribosomal RNA, most of the ribosome, as we'll see, is actually structurally RNA. Okay, and RNA can be catalytic, and actually the catalysis of the ribosome comes from the RNA. It's not associated proteins. We're going to talk about transfer RNA, okay? But then there are a whole bunch of different, you've got small nuclear RNAs, we're going to have RNAi, there's a bunch of other RNAs, if we just continue down here, that have been found where you've got different functions by little pieces of RNA, or maybe not so RNA, but all this sort of stuff, okay? Tons and tons of different ways that RNA gets used in the cell. Probably could do a whole class on it, but we're not going to. Let's see, I need to go one smaller. There we go. But lots and lots of different types of RNA. So we're going to talk here about tRNA, which is, you can think about it as an adapter that it's, or translated RNA, you could, translation RNA, you could think about, it's called that. But it's a piece of RNA that is used to translate the codon on the mRNA to an amino acid. So tRNA is an adapter that, we'll say, connects the codon of a mRNA and mRNA to an amino acid. All right, so if we look at this, what you would have here is you'd have some amino acid attached, and we'll talk about how that is, but you have some amino acid attached up here, right? Off the top of my head, does anybody know what AOC is? AUC? It's not, I don't know which one it is. But you have some codon here, it's AUC, and that corresponds to a specific amino acid, and that's how this gets built correctly. Right, and it's a complicated system, right? We've got a codon, anticodon here, and then you've got some enzyme that needs to recognize this, and it doesn't recognize it based on the codon, and put the right amino acid there. So now we have, if you think about this, if we wanted to change what amino acid got attached, we want to change amino acid associated with a codon, you need to have some sort of mutation here, and in another system over here, and possibly some stuff in here as well, which we'll learn later, okay? Or we'll learn, I don't know if we'll get to it today. But, so, when you think about why does life, when you look at across life on Earth, the same amino acids are used by everything for the most part, right? Why is that the case? And the reason why it's the case that you haven't had some drift there is it's this complex system where you'd have to have, if you just had a mutation here, you'd just break things, right? You'd have, if the tRNA was the same and the codon changed, that you just end up putting the wrong amino acids in, right? You change amino acids in. If you want to change amino acids, you have to change so much simultaneously, multiple mutations have to happen, that it's extremely unlikely. Does that make sense? So one mutation easy, lots of mutations simultaneously not easy. Okay, but this is an adapter, so we're going to read the codon here. We're going to have the amino acid up there. Now you often see these cloverleaf diagrams, but really, this is actually the shape of that. So what we've got is, this is what the 3D shape of a tRNA, where you've got the anticodon down here, and then this is where your amino acid would be attached up here, and then you've got some more things going on. Okay. And these other pieces are going to be involved in, the recognition. Some enzyme is going to attach a specific amino acid here, and it's actually going to recognize pieces in here based on functional groups and slight differences in shape to get the right amino acid on. Okay, the other type of RNA that we're going to talk about is ribosomal RNA. So ribosomal RNA makes up most of the structure and function of the ribosome. All right, so what we see here is all this fuzzy stuff. So the really fuzzy stuff is RNA. So it's left for you guys. At left, the fuzziest parts, we'll say, parts of this crystal structure. It isn't that they don't know what's going on. Is going to be our ribosomal RNA. Okay. The ball version, the space filling there, is actually RNA, is the mRNA. Okay, the wormy bits, okay, it's just showing you the backbones of the proteins. The wormy bits are proteins in the ribosome.
Can you draw arrows actually?
I will. Can I point? I can draw arrows from what I've written. And then the red is the growing peptide. All right, so let's go ahead and quickly, I'll try to quickly in a minute here draw these bits. So the fuzzy parts, let's do fuzzy parts like this stuff. Oh, didn't do it in green. Okay. The space filling parts, so balls, when we think about space filling. The space filling parts are this. Okay. The wormy bits, let's do green. Now let's do blue because they're mostly blue. They're blue and yellow. Wormy bits are here, okay, or here, okay, and then the red is that. Does that make sense? And you can see that there really aren't, I mean, we don't know exactly where the active site is in here based on this crystal structure. I'll probably say it again, we're running out of time right now, but it's like 14 angstroms between where the active site is on the ribosome. There's 14 angstroms before there's any protein, any of the wormy bits, okay? An angstrom is about the size of an atom. So 14 angstroms is a huge difference, and people say that's why we know that the ribosome, the catalysis of the ribosome is actually carried out by the RNA, not the protein. Yes?
The different subunits, like, do they differ based on their, like, is it all still like RNA mostly or mRNA? Does that
Yes, yes. So I'm pretty sure that this is one subunit and this gray is another.
Okay. Thank you.